Hello, so I'm going to start off with a joke. It says, "Please don't sugarcoat it, I'm a diabetic." And to be diabetic means that you either have an issue with insulin production, if you are a type 1 diabetic, or your cells have a problem responding to insulin if you are a type 2 diabetic. So this means that your body has a harder time responding to glucose influx in both cases. And so you would have to watch out for sugar coating in a literal sense if you were a diabetic. Otherwise, your blood glucose levels may get too high and the risk for disease and damage to the body will increase. So let's get started. So today I'm going to be going over how insulin and glucagon regulate the carbohydrate and lipid metabolism. So within the pancreas, there is a region called the islets of Langerhans. And in this region, there are three cell types that are important to peptide hormone synthesis. First, there are the beta cells, which make up most of the cells in the islets and are responsible for releasing insulin. Then there are alpha cells, which are responsible for releasing glucagon. And lastly, there are the delta cells, which are responsible for secreting the hormone somatostatin. Somatostatin is a paracrine hormone that inhibits the release of insulin and glucagon. So, when insulin is released, it stimulates glucose uptake in the liver, the skeletal muscle cells, and in the adipose tissue cells. It also stimulates neuronal signaling in the hypothalamus and acts sort of as a form of negative feedback inhibition in the brain, where it tells the brain, stop eating, no more food, and helps to modify the body's energy expenditure as well as the behavior of the person. So insulin signaling will eventually lead to a loss of appetite. Within the liver, insulin signaling upregulates glucokinase activity, which, along with the increased glucose influx through GLUT2 transporters, leads to a lot of glucose 6-phosphate synthesis. And because of this negative phosphate on the molecule, the glucose 6-phosphate becomes trapped within the liver cell. It can't go back out this GLUT2 transporter. And so it's trapped within the liver cell, so it is either oxidized by glycolysis or it is used in glycogen synthesis. Insulin also activates the pyruvate dehydrogenase complex, which is one of the first enzymes in the TCA cycle. And this leads to the synthesis of more fatty acids due to increased acetyl-CoA production. The activation of protein phosphatase 2A here leads to the activation of acetyl-CoA carboxylase, which is used to convert acetyl-CoA into malonyl-CoA during lipid biosynthesis. Insulin also activates protein phosphatase 1, indicated right here, which dephosphorylates the regulatory enzyme PFK2-FBPase2, indicated right here in the photo I added. So the dephosphorylation of this enzyme activates this portion of the enzyme, and activation of PFK2 downstream affects PFK1. So that is seen right here. Phosphofructokinase 1 becomes activated, meaning the flux through the glycolytic pathway increases. Insulin signaling also deactivates phosphorylase kinase here, which deactivates glycogen phosphorylase. Now, before I move on to how insulin signaling affects skeletal muscle cells, I want to take a quick look into the regulation of glycogen phosphorylase in liver cells. In response to glucagon, phosphorylase kinase is activated. See, so glucagon, phosphorylase kinase. So phosphorylase kinase phosphorylates glycogen phosphorylase. In response to insulin, protein phosphatase is activated, leading to the dephosphorylation and subsequent deactivation of glycogen phosphorylase. This means that during insulin signaling, glucose is not going to be removed from glycogen-reducing ends. Alright, so going back to this diagram, in the skeletal muscle cell, protein phosphatase 1 and the pyruvate dehydrogenase complex are activated just like they are in the liver cells. However, unlike liver and pancreas cells, which have GLUT2 transporters right here on their surface at all times, insulin signaling leads to the translocation of GLUT4 transporters to the cell surface. Glycogen synthase also becomes activated, which means that it will now begin adding glucose molecules to the non-reducing end of glycogen, and glycogen phosphorylase, which removes glucose from glycogen, is inactivated. So back here, this diagram shows the allosteric regulation of glycogen phosphorylase in muscle cells. Because muscle cells don't have any glucagon receptors, they aren't affected by glucagon signaling. Insulin signaling in skeletal muscle cells activates protein phosphatase 1 just like they do in liver cells. And this dephosphorylates glycogen phosphorylase, just, like as shown here. In adipose tissue, GLUT4 the GLUT4 receptor is also translocated due to insulin signaling. This translocation allows for an influx of glucose into the tissue for fatty acids, glycerol, and triacylglycerol synthesis. As you see here, lipoprotein lipase is also upregulated. This protein hydrolyzes fatty acids, allowing for easier lipid uptake into the cell. You can see right here, this is how it works. On the other hand, hormone-sensitive lipase, indicated here, is inhibited, leading to decreased fatty acid release. And then, like I said earlier, insulin signaling in the brain causes you to have a loss of appetite, otherwise known as anorexigenic neuronal signaling. Alright. Glucagon signaling is sort of the opposite of insulin signaling. It is used to stimulate glucose export, as seen here, and fatty acid export, as seen here. Glucagon signaling leads to net glucose export by setting off a signaling cascade, which is done by protein kinase A and CAMP and this G-coupled receptor. And this signaling cascade ultimately leads to the deactivation of glycogen synthesis, the activation of glycogen degradation, and the activation of glucose synthesis. Glucagon signaling in the liver leads to the activation of protein kinase A, which we saw earlier with the insulin signaling and how it affects the liver. So the activation of protein kinase A phosphorylates PFK2 FBPase2. So actually it will be this diagram shown here. The phosphorylation of this enzyme means the FBPase portion of it becomes activated, and this ultimately leads to the activation, or at least the non-inhibition, of FBPase1, which means that fructose 2,6-bisphosphate is activated and can participate in gluconeogenesis. The inactivation of glycogen synthase means that glucose will no longer be added to synthesize glycogen, which is done by the addition of glucose to the non-reducing ends of glycogen right there. And the inactivation of phosphofructokinase 1 means that glycolysis will be inhibited. Protein phosphatase 1 is also inhibited, meaning that glycogen phosphorylase will not be dephosphorylated, as we see here. And that means that glycogen phosphorylase will be activated. So glycogen phosphorylase will be able to remove the glucose from glycogen through glycogenolysis, and basically all of this together culminates into net glucose export. In adipose tissue, glucagon signaling activates protein kinase A, which phosphorylates paralipin and activates hormone-sensitive lipase. Hormone-sensitive lipase then hydrolyzes the acyl ester linkages in triacylglycerides, freeing fatty acids to be used in the energy conversion reactions and freeing glycerol to be metabolized in glycolytic and gluconeogenic reactions. Alright, so just to recap, insulin stimulates a loss of appetite while glucagon stimulates the desire to eat. Insulin stimulates glycogenesis while glucagon stimulates glycogenolysis. Insulin stimulates glycolysis while glucagon stimulates gluconeogenesis. Insulin stimulates fatty acid synthesis and triacylglyceride storage, while glucagon stimulates the opposite, fatty acid and triacylglyceride breakdown.